our first look at cells, we're going to study cells kind of all semester. So you already are kind of way ahead of the game because you know some of the parts and some of the differences. Oh, I forgot one question. What's this? What's this green right here? That's the cell wall. The cell membrane is this little dark green thing right here, right? So that's a cell wall. Good job. Okay, so we're going to be looking at cells all semester, and really what you kind of want to know are the differences between prokaryotes and eukaryotes, so size, structures, and then within eukaryotes, what do each of those structures do, just in general, and some of them we'll talk way more in detail than others in terms of what they do. Excellent. Okay, the next part of today's lesson is on microscopy, which is a little tricky because we don't have microscopes to actually look through. So we're going to talk through it a little bit, and then in your lesson, if you haven't already done it for the week, there's a really cool simulation that you can use to kind of pretend that you have a microscope in front of you and you get to use it. One thing I can promise you is, first of all, everybody in the world isn't using microscopes this semester, right? They're all virtual. And so you will get caught up. The next biology class you take will have you using a microscope. And if you pay attention to what we talk about today, you'll have a little foundation. If you do the simulation, you'll have a little sense of what there is to do. And at some point, I promise, in your biology career, you will have an opportunity to pull out a microscope and actually do what you're learning to do this semester. So it's not like you never are going to get there. You're going to get there. And in some of your cases, you might sit in front of a microscope for years after this, right? So don't panic and don't think, oh, I'm missing out. First of all, hardly anybody's probably using a microscope this semester, especially because they don't want to be sharing them between students, even if they were in person. But you will get your chance. And I absolutely promise that at some point, if you maintain being a biology major, you're going to get your chance. Okay, so what a microscope does is helps us see things we can't see with our naked eye. So if you look here, like we can see a human, we can see some really long cells we can actually see. We can see a chicken egg cell. We can actually see frog eggs. They're pretty little, but we can see them. But most cells that are in these two yellow categories right here, we just can't see with our naked eye. See right here, our naked eye ends and we can't see. So we utilize what are called light microscopes to help us see those. So all those images that I showed you of the cytoplasmic streaming and the pun stuff, right? The cheek cells, those were all under a light microscope. Now, we're going to talk about light microscopes today. There are even more powerful microscopes called electron microscopes that help us see really, really small things like a ribosome. So think about how small a ribosome must be, that if this is a cell, the ribosome is this little teeny dot inside the cell, right? So it's way smaller than a cell. And remember, it's a little teeny dot inside a bacteria cell too, which we know is really small. Can you guys still hear me? I just heard a beep in my ear. Can you text yes? Okay. So, so these electron microscopes help us to to see things that are really small, like ribosomes, or maybe even individual proteins, or even smaller molecules. Okay. Now, when we get to atoms, which we'll talk about soon, that's pretty small. We're not seeing that anymore. We're not seeing that with a microscope. Okay. So again, remember your sizes. Does this fit with what we talked about? Because animal and plant cells are what kind of cells? Can you all still hear me? Okay, eukaryote. So does that fit the range we talked about, 10 to 100? Oh, absolutely, right? And then we talked about most bacteria being kind of right here between 1 and 10, right? So we're right there under 10. Notice, I told you that a nucleus is kind of big. It's the big thing you see in the middle of a cell. A lot of times the nucleus is actually bigger than a bacteria. Isn't that kind of crazy? So the structure inside of another cell is bigger than a single cell by itself. Okay, so microscopes help us to view things we can't see with our naked eye. All right. So microscopy is a technique that we use for helping us see these small things. So maybe with our eye, we could see a field of gazelles or whatever these are. we could magnify it 7 times and kind of see their face, and then we can magnify even more and maybe see a little bit more, okay? So the premise behind microscopy is, like I said, they're called light microscopes, and they pass light through a series of lenses, all right? So the light comes from down here, and it goes up through a whole bunch of lenses and eventually goes to our eye. And people who build microscopes are very careful to build those lenses just right so that what we see gets bigger and what we see is clear and we can see detail, okay? So there are 2 things that are really important when it comes to microscopy. First, the magnification piece, because it's bigger. And then second is the resolution, which is basically the quality or the details of our viewing. So we're going to talk about both of those today. First of all, how many of you have ever used a microscope? Okay, so some of you, okay, good. So you'll probably have some sort of remembrance of what I'm talking about, but like I said, you will get your chance someday, I promise. And I realize some of you, it might have been since like 8th or 9th grade biology, which was a while ago. Okay, so light microscopes rely on light, so just a little lesson on light. So light is not something we can touch, it's not tangible, but it's got these little packets of energy that travel in a wave, right? So if we look at the colors of the rainbow, we could see that the Roy part of the rainbow, the red, orange, and yellow, have a longer wavelength than the Viv part of the rainbow, right? And that just tells us how much energy that light has. Now, this is where the lenses come into play. When light hits different media or different substances, it tends to bend. So let's think about this example. Let's just say you're driving down the street on the hard concrete and you're going 55 miles an hour. All 4 wheels are going the same speed. And then all of a sudden, your front right wheel hits kind of the mud on the shoulder of the road. Now, you're still pushing on the gas. The other wheels are still going 55 miles an hour, but this wheel is going to slow down because it's in mud, right? So what's going to happen to the car when that one wheel starts to slow down? Is the car going to keep going straight? It's going to turn and it's going to slow. It's going to start to slow down, even if you keep your foot on the grass. I'm sorry, not on the grass, on the gas, right? So it's going to slow down and turn the car. And then eventually this other wheel is going to hit the muddy shoulder. So then it's really going to turn the car even more, and it's going to slow down, even if you kept pushing on the gas. Okay, so that's called, well, it's kind of the same in a microscope. So what happens is the light is traveling through air in some places between lenses, and then it's going to hit the lens, and it's going to turn and change its speed, okay? When light bends in this way, we call it refraction. Refraction just means that the light bends when it strikes a different surface, okay? We're going to talk a little bit more about refraction when we get to a technique called spectrophotometry, but just know that that's what's happening. That's the premise behind how that light's moving through the microscope. Now, the first thing we said that's important about the microscope is magnification, all right? So if we look at a microscope, we find that there are, I'm going to go back to this picture, there are lenses that we look through, okay? So those where our eyes go. And then there are lenses right next to the specimen we're looking at. So if we want to take into consideration how much we've magnified, we have to take in the magnification of both sets of lenses. So there are lenses that we look through. Those are called ocular lenses, and we're going to talk more about them in a second. And then there are eyepiece lenses, and then there are the lenses near the specimen. Those are called objective lenses. We have to take both of those into consideration. So to get total magnification, and that's the way we typically talk about it, is we don't just say one lens, we talk about total magnification. We have to multiply those 2 magnifications together. So if, for example, the eyepiece was 10 and the ocular was 10, the total magnification we see would be, what's 10 times 10? Right, 100 times magnified, exactly. Now this is just a really cool picture. This is actually one of those electron microscopes that allows us to see things really little. So this is the tip of a pin. Think maybe like a safety pin, okay? So we zoomed in, magnified it more. And you can see here in this one, like a pin always seems so smooth to us, but when you magnified it, you can see that it's not actually that smooth, and there's these little orange things all over it. Magnified even more to the tip. Look how like not even sharp looking it looks. I mean, a pin pokes us, but at the actual tip, there's still like a flat surface, and you can see these little orange things. And if you zoom in even further, you can see that it's there's a little bacteria. This is why you have to be careful. For example, if you get poked with something really deep in your skin, because it could be bringing bacteria into the deep depths of your skin, which could cause an infection. That's why you have to clean a wound. Now, magnification is only as powerful as the resolution part of it allows it to be. So if you just had a simple magnifying lens, maybe like a magnifying lens you hold in your hand, you could make something look bigger, but the bigger it gets, the more blurry it gets. Can you see it in that picture? However, if in these microscopes, they're built not just to magnify, but to also have good resolution. Remember, I told you that was about the quality. So when you get bigger, you don't just get bigger, you get bigger and you stay clear. So everybody can see the difference between these two. They're the exact same cells, one without good resolution, one with good resolution. So this would be an example of what's called empty magnification. This is just another one. Here's 10 times magnified, 45 times magnified, 100 times magnified. This, you can't really even see what it is. It just looks pixely. And then here you can see with resolution how clear you see cell structures, you see internal structures. Okay, resolution, like I said, is the quality part of this. It's our way of seeing detail in the image. In a way, it's more important than magnification because it's what limits us. We could keep making things look bigger and bigger and bigger, but if it's not clear, it's not useful to us. All right, and resolution is something that's really built into the microscope. So the people, the engineers that build microscopes, they take a lot of time and energy into making the lenses just the right shape so that the light bends just right so that we maximize the energy. However, there are some things we can do on our end to help us with resolution. We can maybe change the angle the light hits it, and that's through a part of the microscope called a condenser. We could maybe change the refractive index so that how the light bends, or we could change the wavelength of the light. Remember how the blue, indigo, violet had higher energy light? We could do that. Now, you do not have to know this math. I'm just showing it to you so you can kind of understand. So first of all, it's not just willy-nilly. There's actually math goes into how they build these lenses. It has to do with the wavelength of light, which is this little lambda character. So remember, we had the red light had a big wavelength and the blue light had a smaller wavelength. There was also the refractive index. So that's the angle at which it's going to bend when it hits the glass. And then it's going to be the sine of that angle, right, that we use. Now, you don't have to know that math at all, but I'm just showing you that it's not willy-nilly. And then we are limited. So this is why I said magnification is kind of not as important, because we can keep making bigger, but if we can't be clear, we're not doing very good. So these are not drawn to scale, obviously. These are lines. These two lines are representing 5 microns apart. These two lines, 2, 1, 0.4, 0.2. If we were to look at it through a microscope, if they were 5 microns apart, we would see them as 2 separate lines. If they're 2 microns apart, we see them as 2 separate lines. When we get to 1 micron, we just barely see them as 2 separate lines. And under 1, they just look like one blob. So in reality, we say that our microscopes are pretty limited to a 1 micron to 2 micron resolution limit, right? We can't see things that are closer together than 1 micron for sure. Okay, the other part of microscopy that's really important has to do with the fact that most cells are transparent. Now, plant cells aren't, they have chloroplasts, but a lot of cells are. In fact, cheek cells, for example, are transparent. You can see right through them. So you can imagine if you have a clear cell on a clear glass with clear liquid on it, it's just a transparent cell. So we need to introduce what's called contrast so that we can see it better against its background. And the easiest way to admit contrast is to dye it. So you can put iodine on it or some other thing. The disadvantage of dyeing it is that you typically kill the cell when you add dye to it. So that's why it's nice to look at plant cells, because you can look at them living and they're already colored. Just a couple of little things. When you look through a microscope, the image that you see is inverted and backwards. So kind of like when you look in a mirror or something like that. So one of the things you're going to do in the tutorial that you're going to go through is you're going to learn the parts of the microscope. So we talked about those lenses you look through that are called the ocular lens. They magnify. These lenses here are called the objective lenses. They also magnify, and there are different size ones, and we're going to look at those in just a second. This is where you put your slide and you hold your slide. There's another lens under the stage right here. That one is more for gathering light. So this is more of a resolution lens. These two are for magnification. This is where you adjust your focus. So there's an outer button or a bigger, wider knob right here that's called the coarse focus. It moves the stage very in big movements. This one's called fine focus. In general, you don't want to use the coarse focus unless you're under really low magnification. Okay, so one of the things I just want to point out is these objective lenses here that you see right here. They're different lengths and they have different magnification powers. So, so the shortest one magnifies 4 times. It has a red stripe. The next one magnifies 10 times. It has a yellow stripe. The next one 40 times. It has a blue stripe. The next one 100 times. It has a white stripe. And you can see that the lens gets longer as it gets more powerful in terms of magnification. But what's interesting is what you see, it's smaller as you go up in magnification. So when you just have the 4x lens in place, now why does it say 40 here if I'm talking about the 4x lens? Anybody want to venture a guess? Here's my 4x lens. Because of the ocular lens. Good job, because that's, if you look here, it said down here, it said field of view at different total magnifications. So this is the 4x times the 10x. Good job, you guys are on it. So when you're just looking at
Hey everybody, I'm back. Can you hear me now? Okay, I'm so sorry. You know what I did? I'm such an airhead. I stepped on my surge protector and I like pressed the red button by accident. Just moved my foot and it kicked it off. So now I gotta move my surge protector. So sorry. Okay, so let me share my screen with you so you can see where I was. Let's see, where were we? I hope the recording doesn't stop. It looks like it's still recording, so. Okay. It didn't, you don't think it stopped? I hope it didn't. It says it's still recording, so hopefully it's still doing it. Okay, so here's where we were. Oh, what was the conversation? Was it a good conversation? Yay, I'm glad you had conversation too. Okay, so what I want you to do when you go into the tutorial, which I'm going to show you in just a second, is kind of learn these parts. Like I said, you're not going to be able to actually use them, but it's probably useful to know what they do so when you get there someday you can do it well. Okay, and then I want you to remember that there's a lot of other kinds of cool microscopy out there. We talked a little bit about the electron microscopy, so allow you to see things in 3D, kind of like this fly eye right here, or 2D like this. But then there's also what's called fluorescence microscopy, so you can label things with fluorescent markers and you can see, so these are labeling different proteins or whatever in the cell, so some of them are red and some of them are green, some of them are blue. Actually the blue is DNA, so that's kind of cool. And then there's microscopes that are connected to computer programs that allow you to kind of take images of slices of a cell, not slices, planes, so it'll go through all the planes of the cell, and then the computer will regenerate it and make it look three-dimensional. So we are only using, touching just a little part of microscopy when we're using a light microscope. Some of the rules of using a microscope that are important that I just want to point out, and at some point you'll have a chance to apply this. You always want to start focusing with the lowest magnification. So you're always going to start with that 4x objective lens, so we are total 40x magnification. Your field of view is way bigger, so you get to see more. And that's when you can use that coarse focus knob. Once you move up in magnification, you never want to use that coarse focus knob because you might slam the stage into the slide. Like in this picture right here, if you use the coarse focus knob, this would slam into the objective lens and you could really damage the lens, which are expensive, or the slide. And then one of the things we didn't point out was where am I? Oh, I don't think I have that version. I'm not in the same version. One of the things that I didn't point out was the 100x objective lens. You have to use oil with it. And so you make sure you only use oil with the 100x. You can never use it with any of the lower objective lenses. Okay. So for this week, your task is to read the lesson. And I'm going to open it up in just 1 second so you can see. There are a lot of videos in there, I think. There are those embedded questions that you have to answer. Make sure part of the lesson is to complete the virtual lab tour. It is really fun. I played with it for a long time. I mean, I'm someone who has used microscopes for 30 years, and I still sat there and played with it. It's actually kind of fun. And then you're going to complete the post-lab analysis. And remember, that's proctored by Proctorio. So you have to have a camera, and it's going to make sure that you're the person taking the test. Some of you asked earlier, can you use your notes? I am fine with you. We always let our students use their notes if we were face-to-face. So I think it's fine, but I want to make sure you're the person taking the test and not someone else. So that's why I still want you to have a camera. Just know when you do quizzes in lecture, you can't use your notes, but just here in this post-lab analysis. Next week, hold on 1 second, I'll answer your question 1 second. So next week we will cover bacteria cells, prokaryotes. In particular, we'll talk about heterotrophic bacteria and photosynthetic, which are called cyanobacteria. We're going to learn a staining process to visualize those cells on the microscope because they're so small, they're hard to see unless you stain them. We'll talk a little bit about how you have to be sterile to make sure you don't contaminate bacteria, and then we'll talk more about that oil immersion. So these are just some thinking questions moving forward in terms of next week. Why is the gram stain important? And why don't we usually use it to do eukaryotic cells? And then how does a eukaryotic green algae differ from a photosynthetic bacteria cell? How are they different? How are they the same? So just some thinking questions moving forward. So you guys had some questions here. Let me answer them, and then I'm going to take you to the lab to make sure we're clear. Is the post-lab just based on today's lesson or also the virtual lesson? Okay, so the lecture we did today and the lesson outline are the entire lesson, right? So you need to, what I covered today is pretty much in the lesson outline, but the lesson outline has a little bit extra that I didn't cover today. So make sure you do both, okay? And then what if we don't have something to record your face? You have to have a camera. So we have a lender program, a device loaner program through TCC. They'll provide you with a tablet that has a camera. So you could borrow that and just use it to take your test. Likely all of your classes are going to require you to have a camera. So I know that in the sciences they're pretty big on making sure that people are taking the tests without cheating and having someone else take the test for them. So that's why we aren't requiring the camera. So if you need a device that has a camera, send me an email and I'll get you in touch with our device loaner system at TCC, and you can just go pick up a tablet from TCC. Okay, so I'm going to take you now, and you can continue to ask me questions, that's totally fine. I'm going to take you now to our lab and make sure that you are doing all of this. So let's do it from student view so that you're not seeing what I see. Okay, so you're going to click on week 1, and you're going to complete all of these things before Sunday at midnight. So you've already done one thing, you came to the live session, yay, come next week at 11 too. But you're going to read the lesson on safety and complete an assignment on safety. And really all it is there at the end of the lesson, there's a certificate that you can upload. So you don't have to do anything, you just have to upload the certificate that says you completed it. And then you can do the lesson on microscopes, eukaryotic cells, and the scientific method, answer all those questions. I think there's 11. And then take the post-lab analysis quiz on eukaryotic cells. Okay, so make sure you do all of those things by Sunday at midnight. All right, I see another question here. What's the difference between the lecture on Tuesday at 12:30 and Wednesday at 6:30? I'm not sure yet. So my original thinking was I was going to do 2 daytime lectures, and they were going to be different. There would be on Tuesday and one on Wednesday, and they would be, additional information. However, when I sent that survey out to you all, I got feedback that some people were not available during the day. They were only available at night. So then I kind of thought, well, maybe I should teach the same thing twice. So I kind of want a little bit of feedback from you. We can cover more if we do 2 different things. If we only do 1 lesson per week, then there's going to be a little bit more learning that you have to do on your own. So if you're someone that can only come to 1, then you will be tasked with watching the recording of the one that you cannot attend. So I would prefer them to be different because I can cover more material that way. But I kind of want your feedback into how you feel. Like if you feel like you never are going to go in and watch a recorded lecture, then that's probably not the best way to do it. Let's see. Lori Ann asks, the week 1 post lab there now. It wasn't there before. Let's see. I think it's one of those things that you have to go through each piece to have it show up. Let me see. It's like adaptive release, like it won't let you have it until you go through everything. Let's see though. Practice quiz. You're right, it doesn't look like it's there. So let me see if I can figure out how to get it there. I will make it sure it's there. Let's see. Maybe it's just hidden. Oh, that's weird. It says item is available, but some students or groups may not have access. All right, I'll figure out why some students and groups don't have access, and I'll make sure you get it. Keep in touch with me if you can't see it. Okay, so I'm done. That was my goal, to be done by 1, so I'm like 3 minutes short, and I turned my computer off in the middle. It's like such an airhead, right? I think I told a lot of you that I've talked to so far in the practice sessions and in emails and in other conversations I've had that I've taught online before, but I've never taught live online like this, where I had live lectures. And I've never taught where I never see my students, because I've always had lab on campus. So I will offer grace and patience with all of you if you offer the same with me. I promise to move my surge protector so that doesn't happen again. But I want this to be a fun semester. I want the learning to be fun. I don't want it to be stressful. I know that there are a lot of things you have to do each week, but just remember, if we were in an actual face-to-face situation, we would be in class for 4 hours a week, and we would be in lab for 3 hours a week, and then you would have to do the additional stuff. So think of it that way, that we're meeting for 2 hours for lab every week, for 2 hours for lecture every week, maybe another 2 hours if you come twice, and then all those other things are just going to go along with it. Any other questions? On the week 1 lab lesson, microscope use, there's a slide to put a graph in our notebook and send a picture in Blackboard, but there's no option to put the picture. Where should we submit it? Let's look at the lesson and see. There's a slide. Remember what page it's on? Put a graph. Second to the last page. This one? This one right here where you click open to folder? Okay, so that wasn't live yesterday. It just went live this morning when classes opened. So if you click on this link now, there is, the pictures are there now. Okay, so that's one thing that's different. Now you're saying you are being asked to take a picture of this and then submit it. Where does it say submit it? I think it just says record it in your lab notebook. I don't think it really asks you to submit it. Does it? In the wrap-up. Okay, in the wrap-up. Let's go there. When you complete this activity, upload a photograph of the relevant page of your notebook in the lab course Blackboard page called Assignment Week 1 folder. Okay, I'll add it. It'll just be called Assignment Week 1 folder. And all you gotta do is take a picture of that table and then upload it. So I'll put it there right after we're finished here. I didn't realize that was in there. I think I didn't go to that last page. Okay, any other questions? All right, so tomorrow we have lecture at 12:30, and hopefully many of you can come. If you can't come, remember that I'll record the lectures and I'll post them. You can always go back and rewatch lab from today. You can scroll through it and see where maybe you missed a little piece, you had to go to the bathroom or something. So if you can't come, try to come to at least one of them per week. Okay, question about extra credit. So for the extra credit we can get, if you form a group and send a picture or video to you, do we just form the groups ourselves and send a video picture, or of our video conference? Yeah, just form a group, find a couple people from in here maybe, and no more than 4, so just 4 or less. And then if you click on the extra credit link in Blackboard, it takes you to a site where everybody can share their camera all at once, so I can see all of you, even if you're at your own homes. And then you just leave it there, and that should work. Oh, okay, the group wiki. Miles thankfully helped me figure this out. I think when you go into your lesson, does anyone want to share their screen with me? Because I can't see student view, so I can't show you what to do. Miles, would you share your screen with me? Okay, I'm going to give you the ability to share your screen. Can you do it? It may be that you can already do it. No, you can't. I got to make you a moderator. Okay, so Miles, now you can, at the bottom of your screen, there's a little like box with an arrow coming out of it that says share content. Do you see that next to kind of the chat bubble? If you click on that, then share your screen and go to your Blackboard page. That's fine. I felt like I saw it for 2 seconds and it went away. Oh, there it is. Okay. So Miles, go ahead and click on our class. All right. So to do the group wiki, on the left menu, scroll down where it says My Groups. And see where it says Group Wikis Text Messaging. Click on that. Click on the, yeah, there you go. All right, and so it gives you kind of the instructions how to do it right here. So you're going to think about muscles and nerves in your hand and what they need to do and how they're organized and how they perform their function. And then you're going to relate those to the themes of life, okay? Now you're going to put your name in red or a color, red, in any color. So Miles might put his name, he has to click up here on Edit Wiki Link. Go ahead and click there. All right, so now he is a typable thing. So Miles, just go type your name in a color right there. So it's that one right there. So pick a color. So he'll type Miles, and then he's going to scroll down. And, oh, you can see Sarah already put hers in. So Sarah and Nancy, you guys had already put answers in, so I went ahead and put your answers in your particular group. So Sarah's blue, so if Miles, if you don't mind, go put Sarah's name in blue up at the top, because she already put her answer in. Okay, so and then so what Miles can do now is he can say, well, I want to talk about theme 1. So Miles, just go down here under theme 1 and just type in orange. You don't just type Miles. You don't have to type anything, an answer yet. Okay, and then when he's done, he can come down here and hit submit. So Miles, you can go ahead and do it because you can go back and edit more later. You can hit submit. Okay, so now if you scroll down, you're going to see Miles's answer and Sarah's answer. So then if Miles wants to add more, or if someone else in here wants to add, go ahead, scroll back up. You can hit this edit wiki content again, right? And let's just say Miles reads Sarah's answer and says, yeah, she's right on, but I think I want to add something. He can go at the end of hers, type in Miles, and then add a statement to say, yeah, you really did good. Let's use the vocabulary ecosystem here because that's really important. So organisms that share ecosystems have to interact with each other. Yes, so this means that you guys are partners, right? That's right. So you get kind of a group grade. The way this is going to work is you all kind of get the same grade. Now, if someone doesn't participate at all, of course, they're not going to get the same grade as all of you. But what I want is it to be complete. So make sure you read what your other group partners are, and then you can add to their answers. How do we know who our partners are? If you go over to the left, Miles, where it says group wiki. I think if you click on that, it should tell you who your partners are. Scroll down and see. Oh, no, maybe not. Maybe it's group homepage. There they are. So Sarah, Miles, and Zane are partners in this group. So you can contact each other, you know, and I know who it is, and you can email or discussion group or something. Okay? So that's how it works. And thanks to Miles, I figured it out. He was my guinea pig and troubleshooted all these different things, but I did finally figure it out. So this is kind of, I would do group work in class where you would have to interact with people in your group and talk. And so this is kind of my way of getting you to talk without actually talking, instead you're typing. And these are due every week, or this is due by Sunday at midnight. That's right. So yes, it's a group thing, and you get kind of a group grade. So as long, you know, Miles and Sarah complete all 5 themes, and Zane never contributes. I don't even know if Zane's here, but if Zane never contributes, Sarah and Miles get the complete grade, and Zane won't, right? But let's just say Sarah and Miles only do 2 of the 5, then they're both going to get partial credit. They're not going to get full credit either. So you need to make sure that you're helping your partners answer the question. Make it more like a discussion. You know, in a real discussion, you would say like, oh yeah, and don't forget this. Well, then you're just going to be typing it like, oh yeah, you're on track, but don't forget this, right? And you're just going to type it in your color.
Are these due like every week, or is it just like a one-time assignment or something?
I'll scroll up and look at the lecture syllabus real quick. It's over here on the left. And then scroll down a little bit till you see like the weeks. So you can see here, oh, go back up a little bit. Week 1 has a group wiki, so does week 2, and then week 3 doesn't. So go ahead and scroll down. And you'll have a different group. Keep scrolling. Like for now, that was all I had. So some of these I haven't actually made my assignments for, so some of these might be a group wiki too, but for now we know we only have 2. No, it won't necessarily be the same group. I'll probably do new groups every time, just to mix you all up, because in a real situation I wouldn't do the same group. Any other questions? You can ask lecture questions too, because you want me for lecture and lab, so that's fine. Just so you know, when we get to lecture tomorrow, there are going to be students in there that don't have me for lab. So if you see new names or something like that, that's why.
For lectures, are you teaching out of the textbook, PowerPoints, or how's it gonna go?
It's going to be a PowerPoint that I'll share. It's already shared with you. If you go to at the end of the lesson, in the lesson for the in the folder for the chapter 1, there's a Google Drive link that'll take you to the PowerPoint. Yeah, you can stop sharing your screen, Miles. Thank you for sharing. How long do you have to get your textbooks? I would say, you know, our first test is in like 3 weeks, so definitely you want to get them, I'm sorry, it's the beginning of 4 weeks, so it's 4 weeks from I would make sure I get it soon, because you really do want to read it. But you know, the lessons and the lectures should suffice if you don't get it right away. The first chapter's, you know, not. It's more of an overview. On the lecture assignment, some of the questions are missing. You mean in the lesson? In the lesson questions? That's interesting. Okay, I'll go make sure that I've made them available to students. I better write these things down that I gotta do. I gotta add assignment, right? For that, for the lab, and then I have to make sure questions are available. They should be, but I'll make sure. How soon we post the lab lectures after they're recorded? It takes a while for Collaborate to give it to me. So sometimes it's a couple hours. So definitely by the next morning, But sometimes, like, I mean, the 6:30 lab's probably not for sure till the next morning, but like today, probably before I go to bed tonight, I'll make sure I check and put it in there. So probably that same day or the next morning. Also, don't forget to add a folder for that week 1 assignment in lab. Yeah, I will. I will add a place for that. And I'm going to check on the questions. Was there anything else I was supposed to do? If you think of something, let me know. I'm scrolling back up. Oh, I guess I can only go to when I turned off. Well, I am so happy to finally get started with all of you. I really want you to do well. I want this semester to be a really challenge, but a fun challenge. So I think we can do it. So if you get lost, if you get behind, if you get nervous, if you get frustrated, if you get, thinking that you're never going to get it, just talk to me. Email me, come to one of my office hours. I promise my goal is to help you get through it. Okay? All right, so I'll see you guys all tomorrow. I'm going to stop recording.